I have great pleasure in introducing the Asian Society Against Dementia webinar, which recently took place on the eve of World Alzheimer's Day. We had the very eminent Professor Robert Friedland of Louisville, Kentucky, talk to us about the gut in Alzheimer's disease. He points out that in 90 to 99% of cases, we don't know the causes of Alzheimer's disease, Parkinson's disease, or amyotrophic lateral sclerosis, and have no truly effective disease-modifying agents. He also points out that despite this, we talk with great certainty. For example, a famous professor said in 2024, the etiology of Alzheimer's disease is now clear. The proximal cause is the accumulation of A-beta amyloid. And while this is true in the narrow concept of disease-modifying agents, it does not necessarily reflect the whole truth and the entire truth. He reflects on his book, 99 Lessons in Critical Thinking, to say, think deeply, don't be satisfied with superficial reasoning. It's important to admit when you don't know something. He asks, is it correct to say that the cause of a traumatic subdural hematoma is bleeding? And points out that there are many ways to approach this particular seemingly simple statement. He goes on to introduce the gut in Alzheimer's disease by pointing out some alternate hypotheses. Our microbiota are the most important environment exposure. Bacteria and microbial products may trigger prion-like protein misfolding as well as neuroinflammation. Alterations in the brain's clearance pathways caused by microbial factors may be responsible. And this lucid talk by Professor Friedland is now available to us on the NeuroKrish Immersion podcast. Do enjoy listening to it. And of course, if you like the NeuroKrish Immersion podcast, subscribe to us and follow us on Apple, YouTube, and Spotify.
We have a very interesting talk today, the gut in Alzheimer's disease, and it's an area we're all very interested in. And of course, we are on the eve of World Alzheimer's Day, which makes it very important both for the Asian Society Against Dementia, which we all represent, and of course the entire Alzheimer's disease movement around the world. Tomorrow is an opportunity for us to remember those who forget and make sure that we don't forget them. And when we think of gut health and Alzheimer's disease, we're all obviously also trying to learn the importance of gut health in terms of preventive neurology and how there is an association between gut health and neurological degeneration as in Alzheimer's disease. Before I hand you over to the speaker, I will invite and introduce our chairman for the day. Professor Vincent Mok is currently the endowed Mok Hing Yu Professor of Medicine and the Master of S.H. Ho College in the Chinese University of Hong Kong. He obtained his MBBS degree at the University of Sydney, Australia, and MD at the Chinese University of Hong Kong. His research over the past 3 decades has focused on understanding the mechanisms of age-related dementia and developing diagnostics and preventive treatments for dementia. He is the first to report the association between cerebral small vessel disease and cognitive impairment in Asia, and he developed the world's first standalone artificial intelligence-aided analysis of retinal fundus photographs for the screening of Alzheimer's dementia and brain health. His major awards include Higher Education Outstanding Scientific Research Output Award in Natural Sciences, First Class Ministry of Education, Excellent Research Award, Food and Health Bureau, 10th Health and Medical Research Fund Anniversary Award, Food and Health Bureau, Chief Executive's Commendation for Community Service, Government of Hong Kong, University Education Award, Collaborative Teams of the Chinese University of Hong Kong, Faculty Education Award, and the Global Australian Award in Education, Science and Research category Advanced Global Australians. He now serves as the honorary treasurer of the Asian Society Against Dementia, which is a very active group of dementia professionals and researchers in the Asian region. And he's co-director of the Charles Cao CUHA Beat AD service. He is the immediate past chairman of the Specialty Board of Neurology of Hong Kong and the immediate past chief of neurology at Chinese University of Hong Kong. Professor Vincent Mok, welcome on to the webinar as the chair, and please do take over from here.
Thanks, Krish. Thanks, Krish, for putting up this webinar for our ASET as a quarterly webinar meeting of ASET. And actually, it's really my immense honor to introduce the speaker of our ASET seminar today. It's Dr. Robert Friedland. He's a renowned clinician scientist in the field of Alzheimer's and dementia. He is currently the Mary and Mason Rudd Chair and Professor of Neurology, Anatomy, and Neurobiology at the University of Louisville School of Medicine. Actually, his work has touched on different aspects of Alzheimer's disease of the last, say, 50 years. My interest is also in imaging in Alzheimer's disease, especially the use of PET. And Dr. Friedland, Rob, is actually the first scientist who reported that with an FDG PET, there's an abnormal brain metabolism in the parietal temporal area, published a few decades ago. And this is really a very important work. Apart from this, actually, Rob is actually one of the first dementia specialists who studied various modifiable risk factors of Alzheimer's disease over the past decades, in gene, in lifestyle, and other things. Actually, recently, Dr. Friedland also actually focused in the role of intestinal bacteria in the initiation of a progression of Alzheimer's and other neurodegenerative disease like Parkinson's and ALS. Over the decades, Rob actually research collaboration extend not outside of US and even to Japan, Middle East, and also to Africa. So actually, it is a real honor that we can now have Rob with us. And as Krish said, actually it is in the midnight to the 3 a.m. in his zone. And we are really grateful to have Rob, Dr. Friedland here with us, sharing with us this a very hot and important topic, the gut in Alzheimer's disease. So we'll pass the time to Dr. Friedland.
Thank you very much, Vincent and Krish. I'm pleased to talk to you about my ideas and work in this area involving gut bacteria and microbes and neurodegeneration. And it's valuable to see how I got involved in this. And it may be related to a visit I made to China in 1987 for the World Health Organization, where I met Dr. Carlton Gajdusek, who of course won the Nobel Prize in 1976 for showing that Kuru and Creutzfeldt were transmissible to non-human primates. Later in Berkeley, I worked with Stanley Prusiner, at least in describing one case of Creutzfeldt. And later, about 15 years ago, I wondered, we know that neurodegeneration can be triggered by products in the gut, as is with mad cow disease or bovine spongiform encephalopathy. In that case, there's something, a molecule in the gut, which is believed to be the scrapie form of the prion protein, gets transmitted through the autonomic nervous system from the gut to the brain. Then you get very sick with spongiform degeneration, and then you die. So I thought, if that's the case, is there any other product in the gut which might be responsible for initiating this process in Alzheimer or Parkinson's? And I found that there are amyloid proteins that reside in the gut that are made by bacteria. And I will talk about those in a moment. And I need to find, how to, yes, so I will talk about how this work developed, what my hypotheses are, what are we doing now, what the future might hold for this. And I will present some of the ideas here in the context of my book, which was published earlier this year on critical thinking, and it's quite relevant to the ideas that have been used by people all over the world in thinking about dementia and about Alzheimer's disease. And it's my conviction that when you're a student, you're judged by how well you answer questions, but in life or in research, you're judged by how good your questions are. And I'm disturbed that in the field we're talking about here, a lot of people have not been asking very good questions, or they've been accepting incomplete answers. The basic problem is, in the vast majority of cases of Alzheimer, it's not caused by genes. And this applies as well to people with 2 copies of ApoE4, in my opinion. And I believe we have no truly effective disease-modifying treatments. The problem with the poor efficacy and great risk of the monoclonal antibodies for Alzheimer's disease is a large topic, and really, I don't have time to go into that in detail, other than the effects are rather small and the danger is quite significant. The fundamental problem is, in my opinion, not thinking very deeply. So Colin Masters, who's a great neuroscientist, says that the etiology of Alzheimer is now clear. The proximal cause is the accumulation of amyloid A beta. I believe this is rather immature or incomplete, because we know many people have amyloid in the brain and they're not demented. Many people die with amyloid in the brain and they're not demented. According to Alberto Espay in Cincinnati, more people die with amyloid in the brain who are not demented than die with amyloid in the brain who are demented. If amyloid was the main problem, then the antibody treatment would be monumentally successful, because it does remove the amyloid from the brain, but it's not that successful. So in regard to my concept of critical thinking, I would like us all to think deeply and not to be super satisfied with thinking superficially. Also, it's important to know and to admit when we don't know something, because frequently knowledge which is incomplete or inadequate or actually incorrect will block our further thought and investigation. Just for example, here's a patient with a right subdural hematoma. Is it correct to say that this is caused by bleeding? I would say certainly there's bleeding, but that's not the cause. The cause is what caused the bleeding. So if he fell off a horse, had a head injury, that's the cause. So amyloid is part of the cause, part of the process of Alzheimer's disease. But I don't believe, I differ in my opinion from Colin Masters. I don't think it's the cause. Alternately, we need to consider our microbiota, which include fungi, viruses, bacteria, and other organisms. We have found, and others have reported before us that there are bacterial and amyloid products, microbial products that can trigger prion-like protein misfolding, and that this misfolding can be transmitted to the brain. My latest work suggests that the alterations in the brain's clearance pathways caused by microbial factors may be responsible, and I'll describe what particular molecular mechanisms I have in mind. Particularly, recent advances in the role of the meninges and the bone marrow in the skull and pathways of clearance of immune cells from the brain is very important and has not received that much attention. Much of this work is from Rustenhoven and Kipnis from St. Louis. Another issue is that the role of the microbiota in the pharynx, mouth, and scalp should be considered. If you look at this figure from a recent science paper, they have completely forgotten about the mouth. They have the gut and the gut as a source of the microbiome. But the mouth has just as many bacteria, or approximately as many bacteria as the gut. And it's largely ignored in microbiome studies, or at least partly ignored. And so we've overlooked the stomach. What else could we have overlooked? Consider neuromyelitis optica, which I will explain in a moment how this is related to dementia. Patients with neuromyelitis optica, which is a demyelinating disease, I believe it's more common in Asia than in the West. Patients with this disease may have antibodies, not always, but they frequently have antibodies to aquaporin 4, which is a water channel protein. So I believe most neurologists would be happy to say the cause of neuromyelitis optica is aquaporin 4. However, I would like them to be thinking more deeply and ask, what's the cause of the antibodies? And we asked that question in more than 10 years ago with Radhika Vaishnav, who's now in India. And we found that there are proteins in plants that are highly similar to aquaporin 4 in the brain. So that exposure to these molecules in plants through diet could cause the production of antibodies, which are damaging the brain. This shows the similarity, especially in D on the right, how the aquaporin in spinach is highly similar to that of aquaporin in the brain. So that of course, we're not supposed to, or we most commonly do not make antibodies to food, but it happens. More recently, Remy Villette, Paul Wilms, and colleagues in Michael Haneke's laboratory in Luxembourg have found that there are also aquaporin 4 similar homologies found in bacteria that could be in the gut. They call it aquaporin Z. And the relevance to the brain is that the aquaporin pathway is thought to be important for the clearance mechanism that goes from the brain and the superior sagittal sinus through the lymphatic and lymphatic pathways to clear amyloid and other toxic products from the brain. And that my idea is if there are antibodies to aquaporin 4, that might interfere with the proper functioning of the clearance pathway. And when you look at this idea, or rather when you look at this avenue of investigation, you find that it's filled with people who are trespassing. We're going from neurology to molecular biology to microbiology. And I think it's quite a wonderful idea to realize we shouldn't be limited by our discipline. This should be, don't be limited by your discipline, and use your imagination. And in 2001, Matthew Chapman, who's in Michigan, found that bacteria make extracellular amyloid proteins. This is a photo of that here, which on the right, which shows highly similar structure to the amyloids in the brain. The amyloids in the brain involved in Parkinson's and Alzheimer and other molecules are pathogenic. These molecules are functional in regard to their production by bacteria. They're made by all the bacteria shown on the left, and they help the bacteria to stick together. And my thought, originally proposed in 2015, was that bacterial amyloids could cause templated cross-seeding of neuronal amyloids, and that process could be triggered in the gut and be transmitted to the brain through the autonomic nervous system. And now, I believe, I don't have that slide, but this was verified or supported by work in the laboratory of Dr. Wang in Hong Kong, who was, I think that paper was published in PNAS several years ago now. Also, there are small bacterial amyloids in the gut, which can be identified. These are all papers that have recently shown some evidence in support of the idea of functional bacterial amyloids. This includes our work from Chen et al. In Scientific Reports 2016. We showed that feeding of bacterial amyloid to aged rats or nematodes caused the cross-seeding, which got transmitted to the brain or damaged the nervous system in the worms. This has also been shown in alpha-synuclein overexpressing mice by Mazmanian and colleagues. And the PNAS paper of Wang was in 2021. We additionally found that E. coli that produce curly make the disease phenotype in ALS model mice, G93 SOD1, G93A mice. It makes them worse. This was the two workers in the study, Simple Kurlawala and Joseph McMillan, shown on the right. And we found that the curly-fed animals were worse on motor function tests. They had more SOD1 staining in the muscle, more autophagy of skeletal muscle, and increased cytokines in the plasma. There was also, I believe, importantly, more astrogliosis in the brainstem, which is a marker of degeneration. And this paper was published in 2023, and I'm sorry to say that it's been cited 7 times, and I believe 2 of those are me or my group. I don't know why there isn't more interest in that, but there hasn't been much work in regard to microbiota in ALS. The most convincing reason why I believe it is important is the question: why does ALS always begin in a certain area of the body? So many people have, the first symptom is difficulty speaking because the tongue is being weak, and other people have it start with problems in the foot. I've asked a lot of people about this. I haven't heard any reasonable hypothesis other than it's random. And that is the explanation that Stanley Prusner gave in his important science paper many years ago, that the misfolding of the amyloid proteins in Alzheimer is random. I find the idea of such an important process being random, I find that very unattractive. However, in regard to ALS, I have a hypothesis there. The reason it starts in different regions of the body is because where the nervous system is interacting with the microbes. If it happens in the colon, that may affect the neurons in the cord, in the lumbar cord, and cause weakness in the feet. If the microbial interaction is occurring in the mouth, it may affect the brainstem first, which would cause brainstem onset, or ALS involving swallowing and speech. So also in Parkinson's, it's found that microbiome-borne curly, the bacterial amyloid, enhance the Parkinson's via the gut. This can be related to dietary fiber, according to Wilms in Luxembourg. But it's also been found that there's more curly in the gut in patients with Parkinson's. Currently, we are investigating tau-22 mutant mice, which is an Alzheimer model, looking at what the bacteria are in the body, in the brain. And we're also looking at the external ear, because the vagus nerve innervates this sensory functions of the skin around the external auditory meatus. And I couldn't find any reasonable paper studying the microbes in cerumen, which is earwax. And this work is being done with Zhangwei Zhu, who is now in Cleveland. We are also looking at complement in these tissues and what bacteria are present in the mice, also in ears and in different regions. Considerable evidence supports the involvement in neurodegeneration of these functional bacterial amyloids. But we don't know which one is most important. So my proposal is that specimens can be obtained from different body regions and studied with detergents and proteases to see which functional bacterial amyloids are present. This could be called negative purification. I discussed this a few months ago with Konrad Bayreuther, who is in Heidelberg, and he thought it was a reasonable question to ask. We don't know. We believe functional bacterial amyloids are involved, but we don't know how much or which ones. Microbiota from the ear can be collected and analyzed in humans using swabs, and this can be done at home. There were only 3 papers with the keywords microbiota or microbiome in the external auditory canal, and no papers about cerumen. And there may be functional bacterial amyloids which have strains, which have particular sensitivity for what pathways or molecular mechanisms are involved in causing disease. I know it sounds silly to mention this, but Alois Alzheimer's dissertation in 1888 in Würzburg was on earwax, and the organs that create microscopic structure of the organs that produce earwax. So it would be kind of some kind of royal justice to find some scientific aspect of that, which of course he was not aware of in 1887. Regarding my interest in critical thinking, the most prescient concept I believe I've seen there is, which I discuss in my book, is this philosopher of science, Paul Feyerabend, who said there is no theory which fits all the facts in its domain. I'm afraid that neuroscientists in general are very smart and they're very good at finding something wrong. Every idea has something wrong. But if there wasn't anything wrong, somebody would have already done it. So if you realize that there is no theory which fits all the facts, we all should be open to the reality that you may have a very good idea and you may have very good reasons why your idea is wrong, which could mean that your idea is wrong, or it could be that what you think is wrong is actually wrong. The best example of that, of course, is Marshall, who got the Nobel Prize for showing bacteria, Heliobacter, cause peptic ulcer disease. And everybody thought he was wrong. I believe he lost his grant. He nearly lost his job in Australia. And he was right. But they thought he was wrong because the teaching was that bacteria don't live in the stomach because it's too acidic. And he was being ignorant by pretending that it did. But of course, he was right because he understood that there is no theory which fits all the facts in its domain. The Nobel laureate in physiology or medicine, Sidney Brenner, also said that he was a great believer in the power of ignorance. You can always know too much. So he believes people who come from outside, who have not been trained into the standard approach, who can see things in a different way. I would hope we could all take advantage of this in our work and in our teaching and make it, and not criticize investigators who have little experience in some area. They may be the most able to come up with new ideas. So I wanted to mention the contribution of my colleagues in the University of Louisville. And this reminds me of my interest in peripheral neuropathy, working with Dr. Martin Brown. Dr. Alan Pestrunk at Washington University in St. Louis showed that there is complement binding to microvessels in peripheral nerves in diabetes. And I asked him why, again, to see how deeply are we thinking about this. Why is there complement binding to microvessels? And his answer was he didn't know. But he had no theory. So my suggestion is it's because of bacteria in the skin which live in direct contact with nerves and nerve fibers. So there are a well-described population of bacteria that live in hair follicles in the skin. And there are nerve fibers that are in direct contact with those bacteria. And of course, this is shown by the phenomenon of goosebumps when the hair becomes erect, which is of course a neural input, which is working on that little muscle in the hair, erector pili, and that is in an area where there are known to be bacteria. So we are working on collecting bacteria from the skin to see if we can find any difference. I would also mention here that I think I didn't mention this. One of the slides had the concept that what difference does it know? What difference does it make to know this? All, in my opinion, that all research should be judged with the question, what difference will it make to know this? So if we know what kind of bacteria are producing bacterial amyloids and how they're interacting with the brain, mechanisms could be devised. There is a company in Boston that is looking at bacteria products to influence bacterial metabolism to treat Parkinson's and also autism. And diet is likely to be related, particularly short-chain fatty acids epigenetically enhance the production of regulatory lymphocytes, which enhance tolerance. And I have neglected to mention the role of functional bacterial amyloids on inflammation, because it turns out that the bacterial amyloids are recognized by the innate immune system by a pathway involving toll-like receptors, CD14, NF-kappa B, and INOS, and the molecular pathway by which the immune system recognizes, through the innate immune system, recognizes bacterial amyloids is similar to that in which the molecular mechanism, similar to that in which the brain's immune system is recognizing amyloids like A beta and alpha-synuclein. I first reported this in the original paper in 2015. Furthermore, with colleagues in Japan, we are working on interactions in Drosophila with feeding of functional bacterial amyloids, and working as well with Dr. Ihara, which I neglected to mention him here, in the Osaka Center, Cerebrovascular and Cardiac Center in Osaka, which is studying primarily stroke. But with Dr. Ihara, we have shown that bacteria in the mouth can influence stroke, particularly bacteria with collagen binding domains can enhance the risk of hemorrhagic stroke, either large or small hemorrhagic stroke in humans. And he's working on treatments for that. Also, the flavonoids may be effective treatments for the functional bacterial amyloids, because EGCG and other flavonoids developed from plants impair the production of bacterial amyloid. And that is a potent area of investigation. I'm grateful for funding from these sources now and in the past. And I will be very grateful for your questions. Thank you for your attention.
Thank you, Dr. Friedland, for the really excellent and inspiring talk. Not only inspiring us on the potential role of a functional bacterial amyloids, microbiota in the colon, in the ear, in the oral cavity, but also the attitude of how we should approach Alzheimer's and other disease as an open mind and to have a critical thinking. Really appreciate it. Thanks. So, any questions from the floor? You can actually, yes, Krish.
Sorry, quick housekeeping. I just wanted to, I've requested people to put the questions into Q&A. And of course, we are a small group of, we can possibly raise hands and get the chairman to allow us to ask questions as well. Can I kick off by asking this question, Rob? Many epidemiological studies have shown rural urban differences in the prevalence of Alzheimer's disease. Do you have any thoughts of what might influence the presence of microbiota in these different environments? How they could potentially how lifestyle or also possibly diet could play a role in sort of putting people at risk.
Yes, thank you for that. Could you show me how do I get back to the page where I could see you?
Just if you can stop sharing, then you'll just go to the screen where we are all talking.
Okay.
Yeah.
Oh, that's very good. That's better. Thank you. The only relationship I know of is that in agricultural chemicals increase the risk for Parkinson's. People who grow up on a farm have a higher risk of Parkinson's disease. And there are important links of Alzheimer's with diet. And like many risk factors involving functional and cognitive and physical activity, diet, smoking, hypertension, and so on. And I have always been perhaps naively suggesting, but not understanding, that in China, the world's greatest public health measure could be produced overnight if the administration would just say, no smoking, and stop the importation and production of cigarettes. I mean, cigarettes have there's no safe use, and there's no good thing about them. There's no function that and if it was made to be unsatisfactory or unacceptable behavior to be smoking, which it is now in the United States, where people are probably embarrassed to be smoking, because in order to smoke in the hospital, you have to walk about a mile away from the door. That's but the problem of agricultural chemicals is that they're similar to products that damage the substantia nigra, pars compacta, and cause Alzheimer's disease. And probably we're all advised to avoid molecules that agricultural chemicals or pesticides or herbicides.
Hey,
Thank you.
thanks. We have a question. It's from Anam from Indonesia. The question is, do gut microbiome change that we see in Alzheimer's disease are also associated with the high burden of Alzheimer's type pathology that is often found in Parkinson's disease with dementia, or even in DLB?
Yes, that's a great question, and we don't know. But the molecular pathways I proposed are basically the same for Alzheimer and Parkinson's. So we don't know why people get Parkinson's disease, but whatever process is going on, it could be it can involve cross-seeding. So the misfolded alpha-synuclein can induce the misfolding of amyloid beta at the same time to create both diseases in the same patient.
Okay.
And this involves amyloid angiopathy as well. CAA can have amyloid in the blood vessel, and this could be something that's triggered by microbial effects. And I forgot to give my 1-minute introduction that our bacteria evolved with us for the past billion years. All of our ancestors had them, and they evolved the capacity to induce tolerance in us, because if they didn't do that, they would be destroyed by our immune system. And our overactive immune system would be very bad for the host, which would not give them a place to live. So the evolutionary pressure for bacteria is to make us healthy. And that is why the bacteria plays such an important role on regulating if we have too much activity, too much inflammation, or not enough. And part of this is regulatory lymphocytes, but this is all pathways which are shown to be dependent in part on diet. The more fiber we eat in the diet, the more relatively tolerance-inducing are our bacteria. And I'm convinced that this is part of Ayurveda, but I shouldn't be not being experienced in this science. I shouldn't be speaking, but I'm sure that Ayurvedic products may be effective in regard to how they act on bacteria, which may involve yogurt and curd, and which have live probiotics, as well as prebiotics, which are food for the good bacteria.
So in terms of, patients often have questions about the kind of diet that puts them at risk of Alzheimer's disease, for example. Vegetarian versus meat-eating diets, and what is preferred and what's not preferred. Based on this knowledge of microbiota, do you have any specific recommendations?
Yes, the general teaching is that meat is absorbed very quickly, digested very quickly, and it doesn't have much food for bacteria. But plants are digested in large part by the bacteria. And depending on how many complex carbohydrates there are there. So that it's very well established that a low diet without meat is protective against colon cancer. And they found that this is related also to inflammation, probably arthritis, perhaps even lumbar spine arthritis, and stroke.
And therefore, as a risk factor for Alzheimer's disease as well?
Excuse me?
And therefore, as a risk factor for Alzheimer's disease, would meat-eating diets, especially red meat, put you at more risk?
Yes, absolutely. And everything about Alzheimer can be linked to vascular changes in the brain, as Vincent has shown so many times. So it's all related. So lack of poor circulation is, and this is all related to our reserve capacities, because the more vascular disease we have, the more sensitive are we to a small dose of Alzheimer, or more able to do well with advancing Alzheimer. Of course, it must be remembered that many Alzheimer patients are quite old, or they're certainly over 75, and regardless of what's happening, it may not affect their behavior until it reaches a certain threshold. And what's really critical is how fast that process goes. So I've advised all my patients, regardless of what happens to them, regardless of what's wrong with them, to manage hypertension, don't smoke, don't have head injuries, Don't let your children play football. Eat fish. Don't eat red meat or pork. And have a diverse diet with lots of different kinds of food, because diversity in diet leads to diversity in bacteria, and that's believed to be the natural state of humans, that hunter-gatherers, which we were until recently, hunter-gatherers had a very diverse diet, much more diverse and much higher in fiber than we have today. So that it's not good to eat broccoli every day. Broccoli is good to eat, but you don't need to eat it every day. You should have other good vegetables at the same time.
Excellent. Really good advices.
Vincent, can I ask one more question?
Of course, Krish. Go ahead.
There's a very interesting recent publication about how a lady could smell Parkinson's disease, and then they attributed to the changes in the sebum, which they could then collect from the neck. And I was really interested in the point you made about the microbiota and the ear, Rob. And I was just wondering whether you see these kind of possibilities developing. Whether a simple swab can actually tell us about our risk of Alzheimer's disease and so on.
That's very interesting. I haven't seen that paper. I don't know, but maybe dogs can be used as diagnostic tools. A dog can probably tell what bacteria are present just by.
I read that in the context of in another context. I think they were looking at sniffing dogs, but this was a lady whose husband had Parkinson's, and she had an extraordinary sense of smell, and she then noticed it. I think she was a nurse. I think she noticed in many of her patients as well. And it then turned out that by doing a swab from the neck, they could actually identify the changes, which became predictive of PD. So I was just very curious that if a swab of the ear could result in better diagnostic prediction, I don't need to give my CSF, or I don't need to have a complex blood biomarker test. So I was just interested in that possibility.
Yes, and you mentioned sebum, that's produced by sebaceous glands in the skin, and it's known that bacteria live there.
Right.
It's described what kind of bacteria live there and live in other places, and so there could very well be a skin product which could be therapeutic or preventive. And I discussed this with Lilly and other companies, and so far they're not interested, but I'm hoping they change their mind. The problem is they're obsessed. They're making a lot of money on the amyloid treatment, the amyloid antibodies. So it's taking up all the oxygen in the room. And the efficacy is about 2 points out of a 70-point scale. And for that, they charge $26,000 a year. I also worry about the diagnostic tests, because the diagnostic tests will only say if there's amyloid in the brain, but that doesn't mean that it's affecting the patient's life.
I think very valuable thoughts on the eve of Alzheimer's World Alzheimer's Day.
Absolutely. So, well, if we have no further question, probably we may end this session, and we will release Dr. Friedland back to his bed.
Yes.
And have a good rest. Yeah. So once again, on behalf of ASAT, I would like to thank Dr. Friedland. Excellent, very insightful lecture, and I must also congratulate acknowledge all your excellent research work over the past decades. Salute to you. And thanks, Krish, for putting up this wonderful webinar. And thanks to every participants of this webinar today.